from the private sector into the government is a great addition.
Thank you very much.
Good.
Thank you, Dave. Morning, everyone. Pleasure actually being on this stage with so many people previously, and I'm sure after are the top of their field from a manufacturing development point of view. It's been a really enjoyable day and a half. I'm going solo. There's no wingman, I'm afraid. I do feel as if I'm presenting from Chelsea Flower Show, which is just down a bit further down the river with all these plant pots behind me. So I was asked to talk to this title, so identifying unique challenges of auto versus allo. And I thought, well, every time you go to a conference, it's always quite adversarial, auto versus allo. So I thought, well, you know, why is it like that? Because there's a lot of common stuff here. So let's start off by looking at the common challenges that we face within the sector, within these two broad fields of cell therapy. So I'm not going to go into the Catapult story. Keith articulated this, extensively on the right-hand chairs here on the opening session. But for those of you who weren't here on day one, the Cell Therapy Catapult, it's a company set up by the UK government primarily it has a vision of making the UK a global leader in the delivery, development and commercialisation of cell therapy. And we want to get businesses who can start here, expand here, and indeed stay here to deliver the medicines that we want and to create economic activity for the UK. We are set up and supported by the Technology Strategy Board, which is the UK's sort of innovation agency, and actually two of the lead specialists are here at the conference if you'd like to speak to them more about how they support this sector very, very ably. So let's just set the scene a little. Auto and allo, where are we in the UK? So at Catapult, my colleagues have pulled together an analysis of the pipeline that we have in the UK. This is available on our website. It was just refreshed recently. And if we look at the UK cell therapy clinical pipeline, that's including products which are 2 years away from perhaps a first-in-man study, we're seeing a nice, healthy growth from 2013 into 2014. Of note from this talk is that we're roughly 2 to 1 a year or so ago, in that we had more autologous and allo, and now it's starting to balance out to become more 1-to-1 ratio. If you break this into the preclinical pipeline only and take the current clinical trials off, you see that actually does become a pure 1-to-1 ratio. So clearly there's a drive coming through for more allo therapies, and that may indeed reflect the maturation of the industry that we all know has happened, moving away from more experimental medicine into more commercially driven outlooks. As a manufacturing developer, what's really interesting for me and exciting is that if you look at where they are, they're mostly in a phase 1-2, phase 2 setting in 2014. This is where we are. They have moved from phase 1s more into phase 2, which is great, but the window is still open here to get your fingers on the process, to start shape what is, to get the commercialization context correct. And to use a phrase which is often used, we're pre-lockdown. We can actually get in here and make a difference. So, just a bit of a preamble there. So, what are the common challenges? We all know in this room we're operating in a commercially driven world. We've moved away from pure scientific interest. I sat on the fence here. I did have moved. I think this room has probably moved from discovery science to manufacturing science, but people outside of this room are still perhaps in this phase. So as a whole, I think we're moving from discovery to manufacturing science, and certainly one of the main activities of Catapult is to accelerate that movement from discovery to manufacturing science. And we've had an awful lot, especially yesterday actually, which was some good dialogue there, is that we've got to manufacture to a sensible price point. And that's going to be one of my core themes here is, how are we going to do that? So, my simplistic view of the world is there's lots of clever people out there who are battling with a health economic issue. That will shape reimbursement strategies. That will deliver a price point. That will give me a cost of good, which I'm told I'll have to deliver to. And then me, my team, and all of our networks, we then have to deliver to that cost of goods. So how are we going to do that? We all know raw material is a huge issue. Very interesting to see Kim Warren's talk yesterday on MSCs, where the cost of goods was roughly about 10%. The raw material cost was about 10%, which I thought was incredible. Whereas, you know, an IPS platform, it's 80 to 90%. Again, it reflects probably the maturation of the MSC field in terms of how much effort has gone into that development so far. Testing, the amount of material that we provide for testing, as well as the expense of actually doing those tests. Yield. A lot of the stuff that we're hearing about from the technology providers is how do we get a better yield? Overhead, well, why are we enclosing? A lot of that's to bring our costs down. And, you know, failure rate. We talk about reproducibility, robustness, ruggedness, all of these things. A lot of that's just to produce our failure rate, again, impacting on our cost of goods. So that's my simplistic, you know, view. I go up there, I see all the bits of kit, get really excited, but ultimately, what it's going to be used for is to squeeze that middle. You know, payers, as we have been talked about the last couple of days, they need to get the best value they can from limited sources around the world. And they go through all these questions. Is the product needed? Is the unmet need there? Is it worth it? Can we afford it, of course? You know, that's the biggest challenge that they're having. Should indeed we control its use, put any restrictions on this? And of course, do we need to fund it? Is there a political imperative or a pressure group to actually push for this? So it's a complex web within that point. Michael May talked about Headspace on the opening day and how they were at CCRM using that analysis to sort of shape portfolios perhaps of where the richer areas to mine are within this sector. And certainly at Catapult we've got a really smart team that's been put together who are looking at that and also in sort of from a different context as well, rather than just a clinical sort of quality of life perspective, but looking at the full value-based pricing model in terms of how that value proposition is being built up, not just from a standard of care perspective, but both the positive and negative differentiation value that goes into those equations. So the standards out there, if you're interested in this, we'd be delighted to talk to you a bit more about that. But let's get back to more simplistic manufacturing items. So we know we're all getting pushed on cost, and that is driving a lot of what we're doing. What are the other common aspects? I've just sort of put it under the umbrella of design for manufacture. I think Phil Vanek, used vein to vein this morning. I used the phrase end to end. We need to understand that complex web. I'll go on to that in the next slide. We had a talk yesterday about quality by design and that toolkit. I've been using this for many years now. I think it's an extremely powerful set of tools. I think it is misunderstood and misused, but the toolkit in there I think is very useful for allowing us to understand this web of raw material complexity. In process control, it's really nice to see people talking about better in process control at this conference and how we're moving from less perhaps reactive processing to more now predictive processing. I mentioned closing in the context of reducing failure, contamination, but also reducing your overhead rate from the AB clean rooms we just heard in the previous talk. But stability, how often do we actually talk about stability, getting new stability solutions? And there are a couple of companies out there doing this, but I think this is a really underdeveloped area in our sector. So when I wake up in the morning, what do I think of about end-to-end supply chain? This is it. We can't work in isolation. We can't think, right, I've got all this complexity in my manufacturing space. I get it in the freezer, that's the end of it. We know that's not the case. There's a huge supply chain around this. And of course, it's not just the physical supply chain of materials, it's the regulatory ecosystem that we bounce into, it's the clinical aspects. And of course, in diagnostics, we started talking about stratified medicines yesterday. Big area, certainly, in immune therapy. just another point I wanted to make on the common sort of aspects is when we're doing the process development and we're putting these things together, is think about it in that end-to-end supply chain. What happens in a process development lab where it's nice and controlled and straightforward often doesn't happen in the real world. So you've got to bring in your validation data sets, latitude and space for failure, I suppose, to be able to deal with the deviations which are inevitably going to occur. So this is some simple data here, which is quite old now, but I think it exemplifies the story quite well of increasing shear down here and processing time here. Fresh cells, which you're going to get off from your nice processes that you're running, and put them through a stressful system, if it's a harvest or a fill finish, whatever it'll be. You're only losing 5-10% of your cells. If you hold it for an hour or so, which could well be what's going to happen when you're in a GMP environment where things are slowing down, you're starting to get into this yellow-red zone where you're losing 30, 40, 50% of your cells. This has got to be worked into your development designs. Okay, so that's, there's a lot there that we're all grappling with, whether or not you're auto or allo. We've all got those same sort of things to deal with. What are the unique challenges? Manufacturing supply model. I think that's the big divergency in this area. And it's often driven by the current technical limitations. Are these models really the models of choice? Are they cost efficient? And I'll just sort of go on to what I mean by this. So, large scale allo, pretty basic. You've got your central manufacturing, you make your stuff, you then move it to the patient. Large batch sizes, long stability, consolidated shipments, etc. Some people have a bit more complexity where they have a secondary manufacturing step. So you're manufacturing in your primary site, you're then doing something which is more than just a basic reconstitution at a satellite site, which then goes to the patient. And in the UK, at the moment, we're investigating to see how we can possibly get a network of these sort of secondary sites set up at or close to the hospitals. It may be that classic pharmacies are not the best route, but clearly there are other cell-orientated facilities within or close to these sites which could be exploited, such as hospital blood banks or NHSBT facilities. And that will allow a model possibly then to be exploited throughout Europe. So, autologous, where do people start? Often because of the manufacturing needs, they often have a central manufacturing point. So there's a lot of autologous material coming in from different sites, coming into a central point, and then going back out with a lot of different logistical movements. That brings in through issues such as traceability throughout the entire end-to-end supply chain. Supply chains get longer and more complex, but Phil said these things which shouldn't phase us. We saw the Walmart data earlier on, and indeed at Catapult we've got a program that we're building up calling Seamless Freight, where we're trying to bring some key providers who are already doing a lot of this together to see how we can grapple with this. Of course, when you're doing this, what's the bottleneck? It's product stability, whether or not it's raw material coming in or finished product coming out. And that gives you your limited geographical constriction on supply. So again, we're being driven by instability, not by perhaps choice in terms of our manufacturing model. So that can often drive people down this local model where you're taking your material, you're doing it manufacturing it at a local site and then going back to the patient. So you have lots of, if you like, parallel sites. So you've got multiple sites to control and manage. That's fine if you've got low product stability. But of course, you're getting a much larger infrastructure cost here. You're getting into product and process comparability. And then, of course, you get to the point of, well, do I have enough sites actually to do this? And just to end on some of this sort of model work, I'm conscious we need to keep to time. You start to think to the future and say, okay, so if we're going to set up these secondary sites, for example, so we've got nice primary manufacturing sites doing your allo, doing a bit of finishing, going to the patient, that's fine. If people are going to invest in their autologous processes, well, those secondary manufacturing sites may actually start to be able to do some primary manufacture, because we've got nice enclosed processes, it's actually a minimal manufacturing input. So it's not the classic manufacturing you have today. It's a pretty hands-on approach. And so you can start to sort of see what the future may potentially look like. So on that sort of manufacturing theme, you know, a lot of people have been asking us about this around the conference, so I just thought I'd put a handful of slides up about this. So we're really excited, incredibly excited, when we got the announcement a couple of months ago now in the UK's budget that we'd also secured an additional £55 million to open up this new large-scale cell therapy manufacturing centre for the UK. And just to put, again, this into context, we've got a really well joined up commercialisation community in the UK. From left to right, we've got a lot of research council money coming in. We've got pre-translational centres that have been set up by the research councils. The Catapult sort of here as a translational centre, doing assay, process development, clinical, etc. Going through then into or being pulled, if you like, into our clinical infrastructure and our reimbursement structure over here. And as Keith said on Monday, we have about 1,000 or so square meters of really good high quality clean room and good quality staff associated with them within this sort of zone, early phase 1-2 centre. So this new centre, which will be run as a subsidiary of the Catapult, it sits up here. So it's taking material from here and pushing it into that phase 3 commercial manufacturing space. Importantly, we're going to operate it as a manufacturing hotel. We've been listening to industry as we were putting this idea together, and there clearly is a lot of interest for people to keep control of their process, not only from a know-how and an IP perspective, but also from a delivery perspective. So it'll be operated as a hotel. This will allow, importantly, contract manufacturers and product development companies to access these core facilities, which will be underpinned by a core quality management system. So people can come to the UK and manufacture their products at large scale. So it's phase 3 and commercial supply is where we're positioned this centre. In the first instance, it'll have 2,000 square metres of production space, housed in a 5,000 square metre or so facility, with all the stuff you would expect in terms of raw material supply and obviously QC on site. And then that will be doubled, or have the capacity to be doubled around about 2020 as we get successful with the centre. Sort of hot off the press is that we're right in the middle of putting a project director in place this week. And we're in a concept designs phase at the moment where we are taking a lot of ideas that we've got and getting that into the operational concept for this facility. We've also put a lot of information on our website this week with FAQs as well, so there's more detail there. And one of the big things with the concept is future-proofing. Again, this has come up in various talks as well. We're sort of open manual, closed semi at the processes at the moment, but clearly we're going to be coming through to close. So how can we address the now and where we're going to be in 10 years' time with this new facility? So, how am I doing for time, Dave? Dave, am I okay?
You have 5 minutes.
Oh gosh, all right. I'm doing well then. So just to close, actually, so I've got a wrap-up slide. I can take some questions if we're ahead of schedule. So to go back to the exam question of auto versus allo, what are the differentiators? Autologous, in my reading of the situation anyway, has just been driven by lack of stability at the moment on your input, raw material, auxiliaries, whatever you want to call them, as well as products. So if we can address that, that will give far more flexibility on people and how they can operate within this space. Something I haven't really gone into for time, but a lot of this results in an at-risk QC release. Real-time data is not available because of the short shelf life of this material, so we're relying on prior validation, etc., and not having full EP tests in place by the time these go into patients. And I think from the data we've seen on an allogeneic perspective, that the costs have been brought down pretty dramatically since over the last 12 months. If you just think about the data that we were seeing last year, Autologous, we haven't seen much data on that the last couple of days, so I think there's going to be a real push with this reimbursement health economics umbrella of how we're actually going to make these autologous products at a price point which is going to make sense for everyone. So again, I think that will mean a lot of energy going into how a decentralized model could actually work. This will probably be driven by a lot of technology in terms of enclosing and predictability, or indeed working with your product so you can put it down a centralized model because you've got a more stable product. So that's it. That's where we are. We're just across the river there, down in South London at London Bridge at Guy's Hospital. There's the website. We've got a stand out there. We'd be delighted to chat with any more questions you have about Catapult or general manufacturing development. Thank you.
I think we have time for a few questions if anyone want to address something to Stephen. I have one. The facility, are you going to kind of all comers? Are you going to look at immunology, the viral immunology, the basic cell therapies, the tissue engineering? Are you going to try to cover the whole spectrum of cell therapy?
Yeah, it's a good question. So the Catapult's remit is to look at all cell therapy, and indeed that's what we're doing on the activities with the current setup. And the new facility will be no different. We'll be taking immune, somatic, tissue to be pushed through that. So that includes gene-modified cells as well. So we'll have the capability to ensure that that class of cell therapy can be serviced through supply of material and be able to handle that material as well. So it's from a design and concept situation. It's interesting.
So we go all the way back into the immunology. Were you looking at developing the lentivirus or the adeno or take your pick?
But it's all about commercial supply. So if it means we've got to manufacture virus in a proportion of this facility as a ring-fenced, clearly operation, then I think that fits within the remit. Are we going to be going back and making novel vectors? I don't think that's the current remit of the Catapult. And there are other centres in the UK and other companies within the gene therapy space and more classic biopharm vaccine space who've got capability and technologies in that area. So this is all about facilitating phase 3 and commercial supply of cell therapies.
Great. Any other questions? There we go.
Can we go back to the area and talk about allogeneic fetal stem cells? Okay, thank you. In fact, I spent many years studying the fetal stem cells. Initially, my inspiration was the work with liver stem cells done and transplanted in Lyon by my friend Jean-Louis Touraine, who did treat the thalassemia and immunodeficiencies in children very effectively. He did it postnatally, in intrauterine, and so on. But anyway, that was my inspiration, and I decided to look for different sources of fetal stem cells, basically bone marrow, because there is a very high contamination of liver cells, from intrauterine environment. Also, these cells are relatively large, and they don't do very well in cryopreservation, and it's much higher loss, and the recovery is lower than fetal stem cells from bone marrow, because bone marrow has very tiny cells, and of course freezing and so on doesn't rupture them so easily. But anyway, what I want to say that, well, this work, I spent many years, and I feel that as far as allogeneic stem cells, this is a source we should really explore, because it is and collect as much tissue as possible and bank them. If I have a poster actually here on that issue. This, as I said, this is partially patented, and I strongly suggest and encourage business to look into this rather safe and large number sources of allogeneic stem cells. They don't express, yet receptors. They are easy to process, and the banking is the way to go in future.
Okay.
Thank you.
Well, I think there's a good advert for your poster there. I think we should all go and swing by. But just on the cryo aspect, I think that's important. Actually, you just talked about large and small. We're running some some workshops with people like the National Institute of Biological Standards and Controls in the UK. They've been doing quite a lot crossover with more pure cryobiologists, and I think we need to get that sort of cross-functional thinking into the space rather than cell therapy people trying to do that sort of thought process themselves.
Thank
Okay.
you.